Hello Achievers, welcome to Team MDS Achievers, myself Dr. Vikram. Today we are here for the discussion on the topic of the biochemical reactions, that is transcription and translation. Most of you people have asked about the biochemical reaction to be explained. See, first thing that you have to know is that the human body is made up of organ systems. Organ systems are made up of organs and organs are made up of tissues. Tissues are made up of cells. Under the cell, they are the biochemical reactions that are occurring. Okay, and there are two types of biochemical reactions that are occurring. Transcription and translation are the reactions that are occurring in the cell to synthesize the molecules. That is, like, transcription is a process where the RNA is synthesized from the DNA, and translation is a process from where the proteins molecules are synthesized from the mRNA. Okay, so, and before we discuss about it, you need to know about why we have to study this concept. See, why the DCI has added this concept under the dental curriculum is because there is certain clinical significance. Fluoroquinolones are the drugs which are the drugs going to inhibit the transcription of the bacteria, so thereby inhibiting the cell growth of the bacteria, right? So without knowing this concept, if you are at all you are prescribing a fluoroquinolone or any kind of drugs, it is like just an RMP doctor prescribing an antibiotics, right? So as a doctor you should be knowing about few of the fundamental concepts. So let us move on, and before we start discussing about the transcription and translation, few of the things we have to discuss, that is mRNA stands for the messenger RNA, that is it carries a message from the DNA to the protein synthesis occurring in the ribosomes. Okay, and whereas tRNA is also called as transport RNA because it transports mRNA from the cytoplasm to the smaller ribosomes. These are the things that you have to remember. Then moving on to the transcription process occurs in three phases, that is chain initiation, chain elongation and chain termination, what we are studying in the chemistry, right? In the chain initiation, to carry out this process, there is requirement of two factors, that is sigma factor and transcriptase is an enzyme which causes the coiling of the DNA, that is uncoiling of the DNA, and thereby the protein molecule or the mRNA is synthesized from it. That is, see, the genetic code that is present in the DNA will be uncoiled, over which the mRNA will be synthesized from one prime end to another prime end. Okay, from where the mRNA is synthesized, and once it reaches the termination codon, that is like once it reaches the termination codon that is present in the particular gene sequence. So what happens? Thereby the termination of the chain reaction occurs. So the mRNA that is produced will be released out into the nucleus, and it exits out from the nucleus into cytoplasm. So once it enters into the cytoplasm, the transcription process has been completed, and we are discussing about the translation process. So this is one of the somewhat confusing thing. So that is why I will explain in a much more storyteller manner so that you will be remembering. Okay. So what happens once the mRNA enters into cytoplasm, it will go and ask the help with the AMP and enzyme. These are the enzyme two molecules which go and attaches to the mRNA and results in the activation of the mRNA. So they will be guiding the mRNA to the tRNA location. Once they find the tRNA location, they will attach the mRNA to the 3' end of the tRNA. See, tRNA has 3' end, 5' end and T-cc loop. For the attachment of the mRNA to the tRNA, 3' end, that is carboxylic group, is the one where the mRNA is attached to it. Okay, so it is crystal clear. So the mRNA is going and attaching to the 3' end of the tRNA. Once this attachment has occurred, the AMP and enzyme says Tata, bye bye, my story has been completed, now you move on. Okay, and they go into the cytoplasm and search for the another mRNAs to do the same activity. So this tRNA with the mRNA will go and bind to the smaller ribosomes. See, ribosomes are two types, that is smaller ribosome and larger ribosome. Smaller ribosomes can be of 30S or 40S, and the larger ribosomes are of size of either it can be 50S or it can be 60S type of ribosomes. Okay, so once the mRNA with the tRNA goes and attaches to the smaller ribosome, which can be of either 30S or 40S type of ribosome, then the larger ribosome will be attaching to the smaller ribosome, and thereby they start reading it. The message that is present from the DNA to the mRNA will be read from 3' end to the 5' end, and then once it reaches the termination region, they will be separated apart, and this as this larger one, smaller one, they will reading it from one direction to another direction, and once the termination has occurred, they will be separated apart, and thereby they are released into cytoplasm. So the tRNA, which has a protein molecule that has been formed over there, and this is released into the cytoplasm. Okay, this protein molecule that is being formed into the cytoplasm is called as primary protein molecule, which undergoes secondary uncoiling and branching chain occurs, and thereby resulting formation of tertiary or quaternary type of protein molecules, and this is the process called as translation, that is synthesis of the protein molecule from the mRNA, and this occurs at the ribosomes, and that's why ribosomes are called as protein factories. And mitochondria is concerned with energy, right? That's why it is called as energy or powerhouse of the cell, right? So that is how. See, this is why we have to study this mechanism, is because whenever we are prescribing certain kind of drugs, these drugs go and inhibit any of these reactions. Okay, and also the attachment of the mRNA, which I said, right, that is mRNA to the tRNA will be occurring at a 3' end, that is 3, which is in a carboxylic group. And this is very important because this was a question that was asked in the my NEET NDS time, that is like in the 2020. So like this, they're going to ask the questions, and also when you are crystal clear with the concept, you can definitely answer it. Otherwise, you will be thinking in the exam that I have studied somewhere I have studied it, and you will be in the confusion and the time will be lapsed. So that's why you have to study the first concept, and followed by you can implicate in the whenever it is required. And why to choose the MDS Achievers Academy is because this is the academy which is majorly based on the one agenda, that is the bridging the theoretical knowledge with the clinical practice, so thereby the success can be achieved. And the main role or the main concept that is involved in the NEET MDS exam is also, right, they are setting certain standards in the dental education that is present in India. That is like a person who is qualifying the NEET MDS exam means he or she has certain amount of knowledge that they can use it for the welfare of the society, is what they are implying. And we are staying on this agenda, that is like both theoretical and clinical practices to be clubbed together to achieve a success, right? And always learn the subject, never ever read or memorize or mug up the subject or concept. Okay, certain terminologies will be present where you have to remember certain terms, but definitely the concept has to be remembered as in a much more story manner. Okay, this is Dr. Vikram signing off.